Good morning everyone. Today we will discuss about the parts of the microscope. Having been constructed in the 16th century, microscopes have revolutionized science with their ability to magnify small objects such as microbial cells, producing images with definitive structures that are identifiable and characterizable. So what are microscopes? Microscopes are instruments that are used in science laboratories to visualize very minute objects such as cells, microorganisms, giving a constructing image that is magnified. Microscopes are made up of lenses for magnification, each with their own magnification powers. Depending on the type of lens, it will magnify the specimen according to its focal strength. Their ability to function is because they have been constructed with special components that enable them to achieve high magnification levels. They can view very small specimens and distinguish their structural differences. For example, the view of animal and plant cell, viewing of microscopic bacterial cell. Microscopes are generally made up of structural parts for holding and supporting the microscope, and its components and the optical parts which are used for magnification and viewing of the specimen images. This description defines the parts of a microscope and the functions they perform to enable visualization of specimens. So ito ang microscope and their parts of a microscope. Meron tayong ocular lens or eyepiece, diopter adjustment, nose piece, objective lens, stage clip, aperture, diaphragm, condenser, illuminator, head, arm, mechanical stage, coarse adjustment, fine adjustment, stage controls, base, brightness adjustment, and light switch. Lahat na ito madidiscuss natin in the next page. So, next page na tayo. Figure, diagram of parts of a microscope. There are three structural parts of the microscope. Example, head, base, and arm. Head, this is also known as the body. It carries the optical parts in the upper part of the microscope. So ito yung siya yung nasa taas kung saan nakakonect sa kanya itong si arm, si nose piece, ganyan. So next, base. It acts as microscope support. It also carries the microscopic illuminators. So, kunyari, pag may titignan ka, ito yung base, diyan mo ilalagay yung kung paano mo siya titignan diyan. So, next. Arms. This is the part connecting the base and to the head and the eyepiece tube to the base of the microscope. It gives support to the head of the microscope and it also used when carrying the microscope. Some high-quality microscopes have an articulated arm with more than one joint, allowing more movement of the microscopic head for better viewing. Wait lang. The microscope arm connects the eyepiece tube to the base. Kung mapapansin natin dun sa picture kanina, wait lang, the microscope arm, sana? wait. Arm connects to the eyepiece. Ayun, naka-connect sila. Gaya ng sabi ko kanina, naka-connect ang microscope arm connects to the eyepiece tube to the base. So, yan. Next na tayo. Optical parts of a microscope and their function. The optical parts of the microscope are used to view, magnify, and produce an image from a specimen placed on a slide. These parts include eyepiece, also known as the ocular. This is the part used to look through the microscope. It's found at the top of the microscope. Its standard magnification is 10 times with an optical eyepiece having magnification from 5 times to 30 times. So, next na tayo. Eyepiece tube. It's the eyepiece holder. It carries the eyepiece just above the objective lens. In some microscopes, such as the binoculars, the eyepiece tube is flexible and can be rotated for maximum visualization for variance in distance. For monocular microscope, they are non-flexible. So, ito yung eyepiece. Kung saan tinitignan, doon sisilipin natin kung ano yung kung saan doon nakikita kung ano yung nasa loob ng in-examine natin. Kunyari, plant cell nga, ganyan, microorganisms. So, next. Objective lenses. These are the major lenses used for specimen visualization. They have a magnification power of 10 times to 100 times. There are about 1 to 4 objective lenses placed on one microscope, in that some are rear facing and other face forward. Each lens has its own magnification power. The objective lenses combine with the eyepiece lens to increase magnification levels. Microscope generally, yun nga, sabi, ang objective lenses and eyepiece lens, combine sila sa isa't isa. So, next. Nose piece, also known as the revolving turret. It holds the objective lenses. It is movable, hence it's called revolve the objective lenses depending on the magnification power of the lens. So next. The adjustment knobs. These are knobs that are used to focus the microscope. There are two types of adjustment knobs. For example, fine adjustment knobs and the coarse adjustment knobs. So ito yung ito yung microscope natin, and nasa gilid yung sinasabing adjustment knobs. Pag ilalapit mo siya, ganyan. Pero meron siyang two types. Fine adjustment knob and coarse adjustment knobs. For the word adjustment, ma-adjust siya kung siguro for para makita mo na mabuti yung nasa loob, ganun. Stage. This is the section on which the specimen is placed for viewing. They have stage clips hold the specimen slide in place. The most common stage is a mechanical stage, which allows the control of the slides by moving the slides using the mechanical knobs on the stage instead of moving it manually. Ito, the stage is a platform. Ibig sabihin, platform siya kung saan ino-hold niya yung mga specimen. The stage typically has a stage clip on either side to hold the slide firmly in place. Some microscopes have a mechanical stage with adjustment knobs that allow for more precise positioning of slides. Yan. So, next. Aperture. This is a hole on the microscope stage through which the transmitted light from the source reaches the stage. So, ito yung microscope. Dito may butas siya. Sabi kasi this is may hole kung saan yun yung aperture allows the light to pass through to the specimen you are viewing. Tapos ito yung stage kung saan sabi, di ba siya yung nag-hold where the object you are looking at is placed. So, yun. Next. Microscopic illuminator. This is the microscope's light source. Located at the base, it is used instead of a mirror. It captures light from an external source of a low voltage of about 100 volt. So, next. Condenser. These are lenses that are used to collect and focus light from the illuminator into the specimen. They are found under the stage next to the diaphragm of the microscope. They play a major role in ensuring clear, sharp images and produced with a high magnification of 400 times and above. The higher the magnification of the condenser, the more the image clarity. More sophisticated microscopes come with an Alb condenser that has a high magnification of about 1,000 times. Sa condenser, wait lang. I-next mo natin yung diaphragm para minsan. Next, diaphragm. It's also known as the iris. It's found under the stage of the microscope and its primary role is to control the amount of light that reaches the specimen. It's an adjustable apparatus, hence controlling the light intensity and the size of the beam of light that gets to the specimen. For high-quality microscopes, the diaphragm comes attached with an Alb condenser and combined they are able to control the light focus and light intensity that reaches the specimen. So, ano kasi, ang condenser lens and diaphragm are condenser lens work together with the diaphragm to focus the intensity of the light source onto the slide containing the specimen. So, si condenser lens, tapos kasi 'di ba condenser, these are lenses. So, si diaphragm and condenser, na-combine sila, work together para makita yung focus the intensity of the light source. So, next. Condenser focus knob. This is a knob that moves the condenser up or down, thus controlling the focus of light on the specimen. Condenser focus knob. So, next. Alb condenser. This is a condenser specially designed on high-quality microscopes, which makes the condenser to be movable and allows very high magnification of above 400 times. The high-quality microscopes normally have a high numerical aperture than that of the objective lenses. So, ito nga. Nakikita natin. Condenser information and numerical aperture. Meron mga nakasulat dyan. May numerical indicator, control, ganyan. So, next. The rack stop. It controls how far the stages should go, preventing the objective's lens from getting too close to the specimen slide, which may damage the specimen. It is responsible for preventing the specimen slide from coming too far up and hit the objective lenses. The rack stop prevents user from moving the object lenses too close to slide.